I'm Dr. Christin Glorioso. I am a physician and neuroscientist, and co-founder and CEO of NeuroAge Therapeutics. And we're an early-stage biotech company that is reprogramming brain aging in order to treat dementia. And this company is based on the last 20 years of my work at MIT and Carnegie Mellon before that. So what I'm going to tell you today, what I'm going to try to convince you of, is that brain aging is a program that leads to neurodegenerative diseases that we can reprogram using either therapeutics or lifestyle interventions. And in a nutshell, what NeuroAge does is we have a consumer-facing product called the NeuroAge test. It can tell you how old your brain is and your future risk of neurodegenerative diseases more than 30 years ahead of time. It's multi-omic and multimodal. And then we gather data from that test, a beautiful longitudinal data set that's one of a kind that we really need to fuel our drug discovery platform for mild cognitive impairment and Alzheimer's disease biomarkers and therapeutics. So we think of ourselves a lot like Tesla. So Tesla is selling cars in order to gather the data for self-driving, and we are providing a consumer-facing product to gather the data we need for Alzheimer's. And before I tell you more details about how we're doing that, I'd like to tell you a little bit about why I started the company. So these folks are my grandparents, and at the end of her life, my grandmother suffered with Alzheimer's disease for 7 years. And my grandfather took care of her in the home. And this had a really big impact on me as a kid, led me to dedicate my life to coming up with better solutions for Alzheimer's. Now, unfortunately, it's in the next generation, so my aunt is late stage with Alzheimer's, her daughter, and I found out that I have a risk gene. So I have a copy of the ApoE4 allele, which is a very common risk gene for Alzheimer's myself. So this is a problem that's very near and dear to my heart. It's also a super common problem. So there's more than 60 million people worldwide that have an age-related neurodegenerative condition. And what we know about dementia is that aging is actually the biggest risk factor for it. So there's an exponential increase in risk of Alzheimer's disease past age 70. And if you're lucky enough to live to be 95, which I think a lot of us in this room will be, people that are very health conscious and have access to medicines, there's nearly a 50-50 chance of having Alzheimer's disease. So it's very common. And the dementia crisis is an escalating crisis. So it's projected to be more than $2.8 trillion problem in the United States. And 50% of those costs are for informal care, so people caregiving in the home for relatives. And a question that I often get is, what is the difference between Alzheimer's disease and dementia? And the answer to that is dementia is actually an umbrella term. So there are many types of dementia that all have some commonalities. So Alzheimer's, vascular dementia, dementia with Lewy bodies, frontotemporal dementia, Parkinson's dementia. And all of these dementias have some common symptoms in common. So problems with abstract thinking, problems with language, memory loss, disorientation, etc. And Alzheimer's disease is the most common type of dementia, which is why people often ask this question, what's the difference between them? The next most common is vascular dementia, and then mixed dementia after that. And all neurodegenerative diseases are different flavors of the same thing. So they are the death of neurons in particular brain areas. So if neurons die in the hippocampus, that's Alzheimer's. If they die in the substantia nigra, that's Parkinson's. If they die in a particular part of the spinal cord, that's ALS. And so actually, in these diseases, you can visualize this loss of neurons on MRI or in postmortem. So brains actually appear shrunken after dementia. And where they shrink is related to what kind of dementia people have. So Dementia with Lewy bodies has a different brain shrinkage pattern than Alzheimer's disease or frontotemporal dementia. And so the question has been forever, why do these neurons die, and what can we do to stop it from happening? So there are actually different causes for different types of dementia, so I'm just showing you a couple here. So vascular dementia is a cardiovascular problem in your brain. So as the name implies, so these are little mini strokes, small blood vessels get occluded or bleed, and that causes tiny areas of the brain to die, and that's progressive over time. Another type of dementia is due to alcohol drinking. So in alcoholics, sometimes get Wernicke-Korsakoff syndrome, which is actually due to vitamin B1 deficiency. And I'm going to come back to the story around Alzheimer's and tell you a lot more about that, since the whole amyloid story has been in the news lately, and there's a lot to think about there. But one thing we do know is that having a timely and proper diagnosis is really important, because some of these causes of dementia are reversible or treatable. So thyroid conditions can mimic it, and there's just thyroid replacement related to that. Vitamin deficiencies can be replenished, and vascular dementia can be treated in the same way that you treat heart disease. So statins, good blood pressure control, etc. And then there's a big question mark around these newer Alzheimer's drugs, which I'm going to get into. But before I tell you a little bit about that, I'll tell you a little bit how neurologists actually diagnose dementia. So the first thing is to rule out vitamin deficiencies and thyroid issues, and to have a brain MRI, and then there's a physical exam to understand whether there's walking or tremor problems that would relate to a Parkinson's diagnosis. And then the most common way that Alzheimer's is diagnosed is through a mini mental status exam. So this is a test of 30 questions. The questions are pretty simple. They're designed at about an 8th grade education level that everyone should get all of them right. So, you know, what is the time, date, month, year, season, and place? So this is just, are you oriented? Can you name objects? Can you subtract 7s from 100? And can you remember 3 words later that were told to you 10 minutes before, etc. And this is scored out of a score of 30. And this is a common example. So a few years back, Donald Trump was actually talking about this exam, the mini mental status exam, with the person, woman, man, camera, TV. So a big question that remains is what causes Alzheimer's disease. So this is Alzheimer's patient, Augusta Dieter. This is the first known case of Alzheimer's, described in 1902. And this woman probably had familial Alzheimer's disease because she was quite young when she started exhibiting symptoms in her 50s. And what this physician did was, after she died, they took a look at her brain and noticed that there were amyloid plaques in her brain. And so from the 1900s, it was thought amyloid must be the cause, these toxic proteins that build up in brains must be the cause of Alzheimer's. And that's led to a whole history that I'll get into. It's controversial. We're still not sure if those amyloid plaques are causal or correlative, or if it's multifactorial. But one thing we did need were some new tools to be able to get at this question. So we needed to be able to understand whether living people had amyloid in their brain so that we could track people over time. So some of the first tools that we needed were PET scans. So in the early 2000s, actually at University of Pittsburgh, where I did my medical training, they developed a compound called Pittsburgh Compound B that could image amyloid in living people. And at that time, there was a professor there who had created Pittsburgh Compound B, who noticed that there were a lot of people that had a lot of amyloid in their brains that were cognitively normal, and so they had no disease diagnosis. So that was kind of the first big question mark with this amyloid hypothesis is if people have amyloid and do not have Alzheimer's, how could amyloid be causing Alzheimer's disease? More recently, in the last 5 to 10 years, we've now been able to measure amyloid in people's blood. So there's a blood test for this. I'm showing you the correlation between blood and brain PET. And this can predict brain amyloid levels pretty well, but not perfectly. And I think that's important because these tests are now widely available. So you can get an amyloid test at Quest or LabCorp currently. So if anyone's interested whether they have amyloid in their brain, this is a pretty good approximation. And again, based on the blood test and also on the PET scanning, there's many cognitively normal folks that have amyloid but no memory loss. But the picture is actually a little bit mixed. So if you have, if you look at this green line on the right, if you have some amyloid, but you also have some cognitive impairment, your likelihood of becoming an Alzheimer's diagnosis is much higher. But if you have amyloid and have no cognitive impairment, chances of you having Alzheimer's in 5 years are pretty low. So there's this sort of mixed picture. So if you get a positive amyloid test, this kind of begins to show you how to think through this. So some other big wounds in the amyloid hypothesis. There was some falsified data. I don't know if everyone remembers some of these headlines. The president of Stanford actually ended up resigning over this. There were falsified gels and images in many publications that had some of the most influential publications around the amyloid hypothesis actually retracted. And then another general problem with the Alzheimer's literature that led up to the current drugs is just a problem with the mouse model. So mice don't get Alzheimer's. They generally die of cancer. And they don't have the same memory problems that humans do with aging. And if you look in the brain of a mouse and you look at genes that change in RNA levels with age and do the same for humans and look at the correlation, it's actually a pretty weak correlation. So there's whole groups of genes that are moving in the opposite direction. So that provides challenges when trying to test drugs in mice, because you need to create a humanized mouse that is pretty different from the natural course of the disease. So all of that being said, there have been 2 drugs that target amyloid that have been approved in the last 2 years. Donanemab by Eli Lilly and lecanemab by Biogen and Eisai. So these are IV medications that are antibodies that do a very good job of crossing the blood-brain barrier and removing amyloid plaques. They slow disease by about 30% on average, but have some bad side effects, including brain bleeding and swelling in about 20% of people. And brain bleeding is a risk to people's lives, so they have to be carefully monitored. It's expensive, and also IV medications are not people's favorite way to take drugs. But I still think it's pretty remarkable that these drugs are working. So the data out of Eli Lilly with donanemab is that 50% of people have zero progression of their Alzheimer's or mild cognitive impairment at 1 year out, which really makes you wonder whether these are going to be worth it for people to take. If I was amyloid positive and had MCI, I would seriously consider taking these drugs. So putting that all together, I think the fact that those drugs work at all means that there must be something to the amyloid hypothesis. So if you remove something that's causal and you see this slow of progression, even if it's not perfect, it does mean that amyloid must be doing something. So my 2 cents on this whole story is that amyloid is a risk factor like cholesterol. So we should be thinking about it differently, not as the single cause of Alzheimer's disease, but one cause amongst many. And Alzheimer's is multifactorial. It doesn't look the same in all people. And so what we really need is better biomarkers and segmentation and to be able to predict who these drugs are going to work for, who they're not. We also need to identify people earlier on because we know that these drugs work much better in people that are very early stage. I think we may even be moving to and Eli Lilly has said as much to the UK government in trying to plead their case to get these drugs approved, that this may actually be a preventative medication in the future, particularly if they can find a way to make these drugs safer. So in the same way you lower your cholesterol to prevent future heart disease, you would lower your amyloid to prevent future dementia. So I think that is the way we're moving. There's also some small molecules that don't have the same brain bleeding and swelling side effects that are in the pipeline in phase 3. So we may end up with safer versions of these drugs that are you can take as a pill. And the other thing that we know is that this better segmentation, so combining A beta, which is the way you track amyloid in the blood, with some other markers, p-tau and NFL, can reduce clinical trial size estimated by 70%, reducing costs. So we really need this way of segmenting people better for these trials. Okay, so this is a long background. I'm going to tell you a little bit what we actually do. So we're working on a whole nother theory of Alzheimer's, a different set of risk factors, and that's aging itself. So many of you have probably heard of the hallmarks of brain aging. So in your neurons and glial cells, you have pathological processes that happen even starting in your 20s. So there's calcium dysregulation, there's mitochondrial dysfunction, DNA damage, changes in neurotransmitter signaling, and specific hallmarks to the brain. So this loss of synapses, the connection between neurons. And then in glia, there's inflammatory processes and glial dystrophy. And upstream of all of those changes are a group of proteins that are master regulators that regulate all of these gene expression changes that lead to the hallmarks of brain aging. We call them BOS proteins. The most famous example of them are the Yamanaka factors that many of you have heard of that can take any cell in the body, turn it back into a stem cell. But there are many other factors and factors that are brain specific that are targetable by drugs and have less of an extreme effect in reprogramming than Yamanaka factors. And when I say program, this is really what I mean. I mean a transcriptional program. So this is expression of 2 genes, but about 10% of your genome looks like this, so over 1,000 genes. So this is RNA levels in human prefrontal cortex. So people die, they donate their brains to science, we get those brains. We can look at the levels of their genes in particular brain areas. And these 2 genes are Alzheimer's-related genes, so calbindin and GFAP. Less calbindin is pro-Alzheimer's disease, more GFAP is associated with greater risk of Alzheimer's. And this is in people ages teenager all the way up to 90s. And what you can see is that we're all being pushed towards Alzheimer's disease by these changes in gene expression levels. And it looks like a program that starts in your 20s. So it's this really beautiful relationships between age and expression levels. And this is just 2 examples. There are thousands of genes that look like this. And they almost unanimously change in directions that would be predicted to cause disease. And but an important point is that some people are actually outliers. So if you look at this person's calbindin level there, they died around age 70, but they had a level of calbindin that was more like a 50-year-old. And we would predict that person to be protected from Alzheimer's disease. So we can combine all of those levels of expression in human brain into a single measurement that we call neuroage. These are machine learning algorithms, and we can very accurately predict how old someone is chronologically. And again, there are people that look older or younger than their birthday age. None of that would matter unless it actually had a relationship to things people care about, which is cognition and risk of disease. So at MIT during my postdoc, we followed older adults for 15 years, and then they agreed to donate their brains to science at the end of that time, and they were followed for diseases, health records, they had longitudinal memory performed. And what we showed is that people that were on average 85 but had a brain that looked 5 years younger, so they were like an 80-year-old, were 6 times less likely to have Alzheimer's in the future. So there's this huge risk factor that we haven't taken into account besides chronological age, besides genetic factors, besides amyloid, which is rate of brain aging itself. And these folks were also less likely to have memory declines. They had better memories overall. They had better moods overall. They had less Parkinsonian signs. So it seemed like rate of brain aging was really a factor for all of these different areas of brain health. And so in order to make this clinically actionable, we had to find a way to be able to do this in living people. So you can't really take a piece of someone's brain that is alive, at least not easily. So we needed to find a proxy for that. So in some of the cohorts, we had both blood and brain. So people donated their brain and also their blood at the time of death. And what we were able to do was create an algorithm that was able to predict their brain ages from a set of 52 blood transcripts. So these are RNA levels. And what this is an organ-specific aging clock. So if anybody here saw Tony Weiss-Corey's paper about a year ago in Nature, he used proteomics and saw very similar things that we do, except we're looking at it by RNA-seq, which is a cheaper, more accessible way to create an organ-specific aging clock. So you go to your doctor every year, you get your cholesterol checked for later risk of heart disease, you get your glucose checked for later risk of diabetes, but no one is checking your brain aging. We don't have a way to do that for later risk of neurodegeneration. So we created the NeuroAge test for brain aging. It's a multimodal, multi-omic predictor. It's comprehensive diagnostic testing. It has those RNA levels, so the brain-specific aging clock, combined with genetics, brain MRI, and cognitive testing online using our software called NeuroGames. We put that together into a super predictor using AI. It's already been trained on more than 500,000 people and is continuously learning from the data we're inputting, and we can give people a brain aging score. So this is Max. Max is 55. He has a NeuroAge of 51. He's 41% less likely than the average man his age to have Alzheimer's in the future. And you wouldn't want this information unless there is something you can do about it. So what we showed is that about 40% of your rate of brain aging is actually due to lifestyle. It's 60% genetic. So there's this huge piece that you can start working on right now to drop your brain aging. And so some of those, we give folks general lifestyle and health recommendations. The big 6 are exercise, healthy eating, sleeping 8 hours, community, which we're all doing here, lowering stress, and staying mentally active. There's also some good evidence in the literature for omega-3s, which are found in fish and walnuts and flaxseed. Coffee and tea are actually very good for your brain aging. That's due to the polyphenols and other antioxidants in them, not the caffeine itself. And then what doesn't GLP-1 do at this point? There's a lot of good evidence for that. And there's been a lot of other studies that we curate and are going to be providing information to people for, both on their dashboard and in our newsletter. And then we also give personalized recommendations. So we have a program called the Sunday Surveys. So we send people surveys every Sunday, figure out what their reported to be eating, the types of diet, if they're fasting, for how long, what types of exercise, what types of therapeutics and supplements they're taking. And then we can combine that using AI with their blood biomarkers to give people personalized recommendations that will work for them in terms of lifestyle. And so I get this question a lot: can you really do anything about this? There's some really great papers that have been coming out in the last 2 years that definitively say yes. So this is a study by someone I went to med school with. He looked at longitudinal brain MRI, and people that had just 25 extra minutes of exercise per week were increasing their brain volume on MRI, so literally growing their brains back, which is quite remarkable. And this is another news story. This is N of 1, so take it with a grain of salt. But this gentleman is an entrepreneur in Florida, and he has very high genetic risk for Alzheimer's. So he has 2 copies of the ApoE4 gene, and was already showing signs of having amyloid accumulation, so even in his 50s. So that was by blood, and then they confirmed it with a PET scan. And then he underwent a very intense lifestyle change, so exercising a lot, vegan diet, etc. Came back and got retested and showed that he was now testing negative for amyloid, which is quite incredible. And then this paper by Dean Ornish that came out in June of this year showed his lifestyle regimen. He'll be at the conference if people are going to the Longevity Clinical Roundtable. He'll be speaking there. So he has a quite intense lifestyle program. It includes diet, exercise, regimen of supplements, and looked at Alzheimer's disease progression and was able to show that people could slow AD progression by 18% just using this lifestyle intervention. So this is a little bit about what our dashboard looks like. This is actually my results. So for HIPAA reasons, I can show you mine, but not other people's. So we give an overall brain age score, which for me is 37. I'm 43. So that's good news. It's more than 6 years younger. And that's a combination of my NeuroGames results. So those are the little games of memory, reaction time, and others. Those blood biomarkers, which is that blood-specific aging clock, and brain MRI, so the volume of my brain. And then how much I have to worry about that is related to my genetic resilience. So I'm only 20% resilient, and that's largely due to that ApoE4 copy that I have. And so I'm pleased with these results. I would like them to be even better, so I'm working on my lifestyle to try to give people even better news the next time I give you this talk. And one thing you can see is that my blood biomarkers and my brain MRI are really closely tracking with each other. And that's one of the things that we're looking at. For most people that have gone through our pilot study so far, we do see that. And what that means is in the future, the NeuroAge test can be potentially just a blood test that's predictive of MRI, which would bring it to many more people and be more cost effective. So a little bit more about what these games of memory are. And I should say that the cognitive testing is actually the least predictive of all of the testing. And that's because people have a really wide range of baseline ability. But this is a game called word pairs. Unlike the mini mental status exam that I showed you earlier, most people get about half of them right. So if you play the word pairs game, don't be discouraged. And you can see this kind of beautiful trend with age. So it's really predictive of our rate of brain aging. People often ask me if this is related to ability. There is a little bit of an education effect. So people with higher levels of education do a tiny bit better. We're partnered with the Stanford Spin-Out Brainky.ai that some people might be familiar with. We had this really beautiful 3D dashboard that we're able to give people that tells you your brain volume and each volume of each of the regions of your brain, and an explanation and some personalized recommendations. We're also running a pilot study that's funded by the Alzheimer's Drug Discovery Foundation to eventually seek FDA approval for the test. And then the ultimate thing that we're doing with all of this information is using this beautiful multi-omic, multimodal data set to identify genes and pathways using causal inference that are the reason that people's brains are aging faster or slower. So some of the markers we're tracking in the blood change with age. They're also druggable. So we've started working on therapeutics pipeline. We're in very early days, but we're working on a small molecule and an antibody that for Alzheimer's and MCI that are brain age reversal therapeutics. We're an expert team with more than 55 years of experience. Sam was my favorite undergrad at MIT when I was a postdoc there. He's now joined the team. Francesco joined the team in September this year. He did his PhD here at the Buck, so it's another wonderful thing that's come out of the Longevity Summit. And then Yuri is our VP of Engineering, and Persiana has come on as our VP of Partnership. She has a background from One Medical and Blue Wave Medicine. And we have an expert advisory board, including Ranjan, who's here, with folks with multiple exits in AI, from training from Stanford, people with pharma expertise, etc. These are some of our current investors. So we went through Berkeley's Accelerator program. We have investment from Alzheimer's Drug Discovery Foundation and Gates Ventures, and then angel funding from MIT Angels and US UK Angels. We're also winners of the National Institute on Aging Startup Challenge, and our lab space is funded by the pharma company AbbVie. This is my last slide. So if you want to try the NeuroAge test, you can try the cognitive games for free for a limited time. That'll give you a brain age score. This is also a 20% off for the full NeuroAge test, which includes the brain MRI and the blood biomarkers piece and the genetics piece. We're having our official consumer-facing launch party tomorrow in San Francisco. You can RSVP at that Luma link, and everyone is welcome to join. And I will take questions.
Wonderful presentation, Christine. Congratulations.
Thank you.
I was wondering if you can, or perhaps your forecast about Medicare covering your product.
Yeah, so we definitely want the test to be covered by insurance, and there is precedent for that. So NeuroQuant, which is partnered with Azra, is just the brain MRI portion, is being covered by insurance right now. So we do think that this could be covered in the future. What we eventually imagine this to be is a blood test like cholesterol testing every year that maybe doesn't have the MRI portion for everyone and these other parts, once we prove out that it's very predictive in longitudinal studies of risk of Alzheimer's.
You said you're like the Tesla, so are you going to use those transcriptional profiles to then create therapeutics that interact with those genes to treat Alzheimer's at some point?
Yes, exactly. So one of the genes, I can't disclose what it is right now, but there's protein levels of this gene go up with age in the blood. And us and other people have shown using Mendelian randomization and causal inference that it's causally related to risk of Alzheimer's more than 30 years ahead of time. So we can track that and see if you have too much of this protein in your blood, and we're creating a therapeutic now to knock down that protein. And we plan to collaborate with pharma on this, so we're in discussions with pharma partners to co-develop. Yeah, so right now we're at sort of early days in understanding the nature of that risk and who's at risk and who isn't. So given the information we have right now, if I was amyloid positive and I started to have a mild cognitive impairment, I would seriously consider taking those drugs. Maybe try lifestyle for 6 months and see if I can work on it that way. If that doesn't work, take one of those drugs and be very carefully monitored. The monitoring is expensive. It's serial MRI. It's also a pain. So that's how I think about it. We know that the drugs have worse side effects in people that have ApoE4, so that's one consideration. They also don't work well in people that are tau positive, so I would say blood testing or PET scanning for tau as well. So these are conversations that families are having with their doctors right now about whether it's worth the risk. If you're very late stage, it may not be beneficial enough to matter, but if you're early stage, it might make a really big difference. Yeah.
Yes. Great presentation, great talk. How much exercise do you need to do? That guy who recovered it completely, is it the 20 minutes a day that is recommended, or do you need 2 hours a day? You know, do you have any comment?
Yeah, so I think, so we know that the sweet spot is around 30 minutes a day, but it's going to depend by person. So that's some of the information that we're going to be gathering with these Sunday surveys. The sweet spot for number of steps is 8,000, so that's about 3 miles of walking or running a day. If you go much more than that, you end up increasing inflammation potentially or wearing down joints, so there's a trade-off with your joint health.
Yeah, thank you very much for the presentation.
Yeah.
Do you envision the microage can be used as a future biomarker, and how is it compare with the current biomarker which the FDA approved? For example, you have a very good critical on the amyloid, which I entirely agree with you, but the FDA approved the drug lecanemab and lecanemab entirely on the changes biomarker, even though it has very little correlation to the actual treatment of the patient. You thought about that, and how can we envision your usage as a future biomarker?
Yeah, we've been thinking a lot about that, and what research I didn't have a chance to show you is that neuroage in the brain is predicting Alzheimer's as well as ApoE4, but it's independent from it, and it's not related to pathology as much, so it's not predicting amyloid pathology, whereas ApoE4 is. So we think it's independent from ApoE4 and from amyloid accumulation. So if you track both neuroage and amyloid, that would make a much better predictor. And we've started talking with pharma companies that have amyloid predictors right now that they're using in clinical trials or to recruit about running little studies to combine the two together to have an even better prediction.
I'm curious from a business and investment point of view, what sort of moat you have around this business, Christin, in terms of global IP or whatever else, or does, on the other hand, cognitive dementia prediction have a risk of being just commodified?
Yeah, so our algorithms are all proprietary behind the neuroage prediction. Also, the data set we're creating with the neuroage test itself will be a significant moat. It's a one-of-a-kind data set that no one else has access to. So that's going to be a big moat for us. And then, of course, in the future with the therapeutics we're creating, there'll be composition of matter and other IP related to that.
Early childhood development in a rich cognitive environment to have a better predictive effect later in age? Is there any data out there? Are you investigating that potency as well?
That's an excellent question. I would predict that would slow down rate of brain aging, but I don't think we have the data set for that yet. But I'd love to start those studies. One more, okay.
Hi there. I'm just wondering if you know much about PBM, the role of red light therapy, etc. I got given I'm using it now actually to wake my brain up because I'm jet lagged, this like red light that you up your nose. And apparently it's meant to shoot in the brain and help us feel alert. But they have published some kind of research on like, slowing cognitive decline. I just wonder if you knew anything about it.
Yeah, so I took a very quick read of the literature around red light therapy, and as far as I know, there's some evidence, particularly in wound healing. I haven't read the Alzheimer's studies myself, so I can't really comment, but I think there is potential promise with red light therapy.